Hey there, folks, and welcome to Chapter 9, The Patterns of Inheritance. This video will cover sections 9.1 through 9.10. First video. Chapter 9 gets us into the basics of genetics. Now, the human interest in genetics is actually a very old one. It dates at the very least back to the ancient Greeks and quite possibly before that as well. What we are most known for in basic genetics, however, is the system of study established by a monk named Gregor Mendel. Mendel studied mostly out of necessity. He was a 19th century, 19th century, that's still pretty recent. He was a monk who specialized in his garden at his monastery. And by studying his plants, he realized that certain aspects of his plants were heritable. Now, heredity means that certain traits are passed down from parents to their offspring. And by studying his plants, Gregor Mendel identified some heritable traits that he was seeing in his favorite plants, his peas. He realized that certain plants would have green peas or yellow peas or constricted pods or nice round inflated ones. He realized that he could control how tall the plants became and a number of other genes and other traits as well. And from his notes, he developed certain theories about what has become the modern field of genetics, the study of heredity, these traits that are passed down from one generation to another. And Gregor remains known as one of the many fathers of modern-day genetics. There's some terminology that you should know before we get further on into genetics, just to help save some time. When we look at a combination of two different varieties, it may be, say, when we mate one flowering plant that has red flowers by, say, another one that has white, the resulting offspring are called hybrids. They combine, or they potentially combine the traits. The hybridization is called a cross. You are combining their genes. You are crossing them. P generation is shorthand for the parent's generation. F1 stands for their first generation of offspring. F in this case stands for the word filial. You can have an F2 offspring, which would be the grand offspring of the original parents. You can have as many F generations as you need to for the course of your study. What Mendel was doing back in the 1800s was studying his plants one trait at a time, which is something called a monohybrid cross. He was looking at one set of genes at a time. And in doing so, he realized that different versions of the same trait react with each other in fairly predictable ways, and that he could even use this knowledge about how they interacted with each other to predict what sort of offspring different parents would have. When we talk about different versions of the same trait, those versions have a specific name. They are called alleles. Say, for instance, we're looking at a fairly common trait like hair color. We all have genes for hair color, but plainly there are many different colors of hair. Brown, blonde, red, black, anything in between. Each version of hair color is a different allele. So, say, even though two separate people are carrying the same genes that code for hair color, they may have different alleles, and one may be blonde, and one may be brunette. When our alleles interact with each other, not all alleles are created equal. Your body will use certain alleles instead of others, and this is because some alleles are dominant and others are recessive. When our cells use our DNA, the genes that actually get used and expressed are the dominant alleles. The moment a dominant allele is present for a gene, that is the one our cells use. However, the recessive alleles can still be used. You just need a very specific circumstance in order for that to happen. This is called the genotype. It is the combination of alleles that an individual is carrying for a specific gene. When the genes are the same alleles, we each get two different versions of a gene for most genes, one from either parent. When we've received the exact same allele, say we got the dominant allele from both parents, when they are the same, this state is called homozygous. There are two different versions of homozygous because we could receive two dominant or two recessive alleles. So when we are looking at genotype, if the case is homozygous, it is not enough to just say it is homozygous. We have to specify which one. Homozygous means that the two alleles were the same in the zygote. Homo means the same, zygous refers to the zygote. So if you ever have a hard time remembering these terms, there's one way you can break it down and it will help you remember. If we got different alleles from either parent, and we have one of each, one dominant and one recessive, this state is called heterozygous. Hetero meaning different. There's only one version of heterozygous because you have one of each. We never specify whether the heterozygous is dominant or recessive because we have both present. The way a recessive allele actually gets used is if there is no dominant allele to cover it up. So out of the different combinations of alleles I've just discussed, the only one that will get you a recessive trait that's actually being expressed in any living thing is homozygous recessive, where both alleles are recessive and there is no dominant allele there to cover it up. When the genes actually get used and expressed, the resulting physical trait is called the phenotype. So say we were to discuss, well, how about adorable red hair, as in this XKCD comic? Red hair is the recessive trait for a specific, expressed by a recessive allele for the hair color gene. All right? The recessive allele for the hair color gene is in the genotype. It's encoded in the DNA. But what happens when those genes actually get used is adorable red hair. Needless to say, just talking about this can be a little confusing. So geneticists have tools to help describe and diagram out exactly what these relationships look like. This tool, our best tool for this, is called a Punnett square, and you're going to love it. It is so simple, it is so useful. Let's have a look. We're going to first do this with peas because peas are what Mendel was studying. One of the traits that Mendel looked at in his garden was the color of his pea pods. Some were plainly more appetizing than others. In his pea pods, green was the dominant color. And when we do a Punnett square like this, when we're handling the genotype, our dominant trait is always symbolized by a capital letter. Yellow was our recessive, which we symbolize with a lowercase letter. In order to run a Punnett square, you basically draw a cartoon drawing of a window or just a large hatch mark. Either one will work. At the top, you place the letters symbolizing the genotype of one parent. So what are the letters symbolizing the alleles that that parent inherited from their parents? The other parent goes on the column to the side. In order to finish running the Punnett square, because now it's all set up, you just take those letters down and across from each parent. Dominant letters go on the left, with only one or two exceptions. So these two plants each are homozygous, meaning both of their alleles are exactly the same, but one is homozygous dominant and the other one is homozygous recessive, have 4 heterozygous offspring, or they would hypothetically. Now, you can do this for anything. You can do it for pea pods, you can do it for people, you can do it for fictional characters, with some interesting results. When you are looking at basic genetics, when you're especially doing a Punnett square or looking at the genotype for an individual, what you're actually investigating is what specific allele was inherited from their parents in their DNA. Right? When one gamete meets another and an egg is fertilized by sperm and a zygote is formed, all of this DNA from each parent is combined. And so on specific homologous chromosomes, referencing back things that we covered in a previous chapter, you, for instance, received 2 different alleles from each homologous chromosome that you inherited from your parents. There are specific loci, plural of locus, which are where these alleles for these genes are located. For some traits, we actually know exactly what chromosome that allele for that gene occurs on, and we can physically locate it. For others, there's multiple genes that may manage a single trait, and so the loci for the alleles aren't quite so easy to pin down. What Mendel did is a fairly simple study. His observations follow the basic laws of probability. He could even, because of the behavior of the traits that he was looking at, he could even track 2 traits at the same time and saw that one trait did not affect the inheritance of the other. They were inherited separately. This is what the law of independent assortment is. Just because, say, one plant may contribute genes for yellow pea pods does not affect how well it can contribute genes for, say, a nice round pea pod, as opposed to a constricted shriveled one. A test cross is the process of doing a Punnett square in reverse. Say, for instance, you had the hypothetical offspring of 2 people, but you did not know what the genotype was for one of the parents. By looking at their offspring's genotypes, you could work backwards and figure out what that missing genotype was. So this Punnett square is simple to set up, it's simple to run, and you can even run it backwards. It's just as easy. For instance, you can also use a Punnett square to calculate what are the odds that a specific genotype will come up if 2 individuals had offspring. You can do this 2 different ways. The first is the law of multiplication. Simply by multiplying the half of one parent's genotype by the half of the other parent's genotype, you can figure out in a simple small Punnett square like this that, say, the odds of getting a particular genotype combination might be 1/4. Or if it's a very common one, as in the case of this Punnett square, you might even get 1/2. Working backwards, say if you wanted to do a test cross, you could use the rule of addition and add up the likelihood of getting particular offspring from a specific set of parents, according to what the offspring already are. In order to track all of these over time, because again, you can do multiple generations, we use a type of diagram you might already be familiar with. It's called a pedigree. If you own a cat or a dog, you may need to track your pet's family pedigree. Many of our pets have specific genetic diseases. that can stay in the family. And by knowing what genes a father or great-grandmother of our pet carried, we can predict how healthy our pet will be over time. That's actually what a pedigree is all about originally. Now, sometimes it's for tracking purity for a breed, but originally it was for tracking useful traits and health, such as this pedigree for a line of horses. As you can see, every single parent has marked down what their genotype was for a particular trait. Just because some of those traits may be particularly common, though, doesn't necessarily mean that they were dominant. In wild-type traits, these are the most common traits that we see in nature. The dominant trait is not always the one that we see most often because of ways those traits may affect an individual's survival. There are also some recessive traits that can stay in a population, even if they are less useful, so long as they don't affect the survival of the individuals that have those recessive disorders. The disorders can stay in the gene pool for a population. One of the ways this can happen is because the trait is recessive, it is possible for a parent to carry it from one generation to the next without actually expressing that trait themselves. So long as the parent is heterozygous and they have one version, one allele of each type. Remember, if it's a recessive trait, that dominant allele is going to cover it up. That parent won't actually use the recessive allele, and so they'll remain healthy and without the disorder. But it's still in their DNA, and they can still pass it down to their offspring. This carrying it down to the next generation is why these individuals get called carriers, and it only works for heterozygous parents passing down recessive disorders. An example of this is color blindness. Men are much more likely to be colorblind than women because the gene for colorblindness occurs on the X chromosome. Men only get one X chromosome. Women get two. So women get two chances to get the dominant allele, which would cause them to have normal color vision. But if a woman were to end up with an X chromosome with the recessive allele for colorblindness from one of her parents, she could pass it down. She'd still have normal color vision herself, but her children might need glasses that would give them color vision like you see here on the right. The picture at the top of the diagram shows an estimation of what it's like to be colorblind. But now, due to technological advances, we have special sunglasses that can help colorblind individuals actually see in color for the first time in their lives. One of the complications with genetic disorders, however, is that not all of them are recessive. Some of them are dominant. A good example of this is Huntington's disease. Huntington's disease is a nasty piece of work, and eventually it can be fatal. The only way it manages to stay in a population where it eventually kills the people that carry it is that Huntington's disease typically doesn't start to cause trouble until someone is in their 40s, at which point, if they're going to have children, they frequently already do, and the gene's passed on already. To round out this video, let's talk a little bit about technology. We were just talking about those really awesome new sunglasses. Hop online. There are videos of someone's experience when they first put them on and see the color red for the first time in their lives. It's amazing. That's not the only thing that we can use genetic technology for. We can also use it to protect ourselves. There are a number of different ways to run genetic tests, to test for a particular disorder or gene presence in our DNA. We can also do it for our children, even as early as before they are born. We can test the amniotic fluid around a fetus in the womb. We can also test a little sample of the placental tissue. That's what chorionic villus sampling is. Testing the fluid, by the way, is amniocentesis. For fetal imaging, to look at how a fetus is physically developing, we can use an ultrasound. We can also do a whole other series of newborn screenings to make sure that a newborn baby will be healthy. However, just because we have all of this technology and we can use it, does not mean that we always should. And with the development of all this technology also comes a new suite of questions about when and whether it's always ethical to do so. That is a long-running debate, and medical physicians have to balance very carefully their interest in protecting the health of their patients and their patients' newborns, and whether or not it is ethical to screen for certain genetic disorders. All right, folks, that's going to wrap it up for this video. I know this is a little bit longer one with some technical jargon thrown in there. Well done for hanging in there. I'll see you next time, folks. Bye.